Hello. As always, thank you for being here this early. Hi, Anna. Hello, those in 10 Evans. Today's lecture covers what is probably at the heart of the discipline of genetics. And it physically hurts me that the work of Gregor Mendel, the shining titan of this field, is, from my perspective, consistently mistaught. The moment somebody says Mendel, you go, oh, not the peas again. Oh, yellow versus wrinkled. Ugh. So I want to do everything I can to bring Gregor Mendel's work to life for you, and I'm going to do so using a rather overwhelming example, and I accept the fact that some of you might get overwhelmed by this, but reality is reality. The New York Times in 2007, and the article is still online, so I encourage you to read it if you have time. It's not required, I just encourage you to do so. Wrote an article about a woman named Deborah Lindner. At the time she was 33. I actually had the pleasure of meeting her via a Skype conference recently. She's a physician. The article is entitled Cancer-Free at 33, but Weighing a Mastectomy, which of course is a medical procedure, which is surgery to remove the breast. Deborah Lindner, says the article, did intensive research as she considered having a preventive mastectomy after a DNA test. Why would somebody have a preventive mastectomy? The article continues to discuss the implications of a finding that in fact was made here on the Berkeley campus by a geneticist named Mary Claire King. And she found the reason why of the approximately 45,000 women that die in the United States every year of breast cancer, in approximately 5% of the cases, the cancer is hereditary. And that has to do with a mutation in a gene called BRCA1. And in fact, in Deborah Lindner's case, she knew from her family history and then subsequent genetic analysis that she was a carrier for the mutant form of this gene, and she therefore knew that she has a lifetime chance of approximately 70% of developing essentially incurable breast and ovarian cancer. And she knew this because her mom, Joan Lindner, had breast cancer at age 48 and then tested positive for the gene. Dr. Lindner herself went ahead and had the analysis done. Her sister, incidentally, tested negative for the gene. I hope this will work out. I'm not sure. I tried this earlier. On the New York Times website, and there's a 5-minute video which I don't have time to show you in its entirety, is a truly remarkable video of showing Dr. Lindner preparing for the surgery. There is this 33-year-old woman brushing her hair, you know, preparing for this radical surgery, which she went ahead and had done. And she's alive and well and just fine, and of course disease-free. You know, honestly, I'm not a warm and fuzzy person, but I find it hard to watch this without sort of a lump in my throat. It's kind of overwhelming. There she is after the surgery. If you have a very strong family history of breast cancer, I'm not a physician, but you should be aware of the fact that the option of getting tested for whether or not you have that mutation is available to you. You should not get tested without first carefully considering the implications of finding out the results of your test. And I'm not a physician, so this is not advice. This is just a statement. Why am I telling you this? Well, if you read the article further, it says, generations of the Price family have been affected by a mutation in the BRCA1 gene that significantly raises the risk of breast and ovarian cancer. And then there's a statement that describes a general principle. And the statement is, a parent who carries the defective gene has a 50% chance of passing it on to his or her children. In fact, the Lindner family is a perfect example of the truth of that statement. Joan Faith Lindner has a 50% chance of passing the mutant form of the BRCA1 gene to her children. One of her daughters, Deborah, acquired that mutant form. The other one of her daughters, Lori, did not. By show of hands, can somebody tell me what the name is of the person who discovered this scientific principle? Okay, let me show you that person. It's Gregor Mendel. Here's his garden in Brno in the Czech Republic. It's still there, where he made that discovery. The principle that you just saw, that a parent has a 50% chance of passing a mutant form of a given autosomal gene to his or her offspring, is known as Mendel's first law. And my purpose today is teach it to you, such that you will remember it forever, because I think it's one of the more important principles, not just of biology, but of science in general. Part of the disservice that is done to Mendel in teaching his work is, in retrospect, it seems so obvious, so almost trivial. To try to dispel that concept, I need to put Mendel's work into a little bit of context by showing you studies on the problem of heredity performed by two of Mendel's predecessors. And I am deliberately not picking random people. I am picking absolute titans of science. So the greatest scientists of their day. This is a painting by Jan Vermeer, the great Dutch artist. It's called The Astronomer. It actually is in the Louvre, another museum I encourage you to visit. If you're ever in Paris, go to the Louvre. It's well worth it. It is thought to show Antony van Leeuwenhoek. Now, Leeuwenhoek, in fact, did not invent the microscope. He built the first spherical lenses that worked really well. And in fact, this is one of Leeuwenhoek's original devices. It still works beautifully. You can take a majestic picture of human red blood cells with it. So Leeuwenhoek, incidentally, as some of you know, in VLSB, there's a beautiful exhibit of old microscopes, and there's a copy of Leeuwenhoek's original book in there. I really encourage you to check it out. It's actually quite remarkable. Leeuwenhoek was brilliant, and as many scientists of his day and today, was a polymath, so he was interested in everything. He decided to investigate the problem of inheritance, to find out how traits are passed from one generation to another. And he did the following experiment. He decided to study the inheritance of coat color in rabbits. He was surprised to find, as this review says, and if you have time, read it. You don't have to, but it's very well written, that a gray male wild rabbit gave rise only to gray offspring. Okay, well, what is the conclusion from that? Well, here's Leeuwenhoek's conclusion. His conclusion was that sperm were the sole source of the future animal. Quote, a proof enabling me to maintain that the fetus proceeds only from the male semen, and that the female only serves to feed and develop it. In other words, there was no relation between both parents and the offspring, but simply between father and offspring, which was represented by the little animal in the male semen, which Leeuwenhoek thought he saw using his remarkable lenses. The review continues, and realize this was in the late 17th, early 18th century. It is tempting to imagine that if Leeuwenhoek had done the reciprocal cross, using a gray female wild rabbit, or if he had studied the grandchildren of the gray male, he might have paused for thought some further, and the course of science might have been changed. But he didn't. He was convinced that sperm are the carrier of inheritance, et voila! He did an experiment that, quote, proved it, unquote. A century later, it got no better. My favorite example in that regard comes from probably one of the greatest naturalists of all time, the Frenchman Comte de Buffon. He was fascinated with the problem of animal hybrids. You know, like the wolfen or the zorse. He chose to work with quadrupeds, so four-legged animals. It turned out to be difficult to do a controlled cross. For instance, during Buffon's attempt to mate a wolf with a dog, the female wolf ate the dog she was supposed to mate to. Now, just imagine the dialogue. Well, how did the cross go? What was the result? Oh, Monsieur le Comte, the dog she ate. Just let me reemphasize. These were not quacks. These were not vendors of snake oil or, you know, of collateralized debt obligations. Oh. This was the foremost thinker of his time, and he would do an experiment in which he would try to mate a dog with a wolf, and the wolf would eat the dog. Yeah. And into this steps a man from remarkably humble belongings. You know, Newton, Darwin, and Einstein, and Mendel, most people agree, is one of the only people who is worthy of joining that remarkable lineup. Was the only one from truly humble beginnings. So talk about a Horatio Alger story. Probably should be called a Gregor Mendel story. He was born in a peasant family in Moravia. He showed exceptional promise as a child, and his sister gave up her dowry so that he could go to college. And then in a lesson to all people of all time, he went to the University of Vienna and he studied no less than with Christian Doppler of the Doppler effect, of the redshift, of all of that fame. So he learned his math and his physics from a titan of science. He had to drop out of college. He got nervous during exams. And he chose to then join a monastery, and he became a friar, not a monk, a monastery, because it was quiet, and he could do his work in peace and solitude, and he chose to breed peace. And he achieved immortality akin to that only 3 other scientists have. So if you feel nervous during the midterm, say to yourself, just like Mendel. Incidentally, Woody Allen, of course, famously said, "I don't want to achieve immortality through my works. I want to achieve immortality through not dying." That's not likely to happen. However, this, oh wow. The history of biology knows only 3 publications of this caliber. Of course, Charles Darwin's The Origin of Species, James Watson and Francis Crick's 1953 paper on the structure of DNA, and Versuche über Pflanzenhybriden, Studies on Plant Hybrids, a detailed paper published in an obscure naturalist journal in 1866. It is truly beautiful. I am a passionate basketball fan, and I had the good fortune of seeing Michael Jordan play. I think this is, in my mind, second only to that in terms of just beauty. It's just like, or those of you who are fans of football, it's like watching Lionel Messi dribble. It's just, oh, Mendel's paper is beautiful. What did Mendel discover? I'm going to use modern day nomenclature. Mendel discovered no more and no less genes. Furthermore, as if that wasn't enough, he discovered 2 basic universal principles according to which the genes are transmitted from generation to generation. We now know, he didn't know it at the time, that the principles are universal because they reflect core mechanisms of meiosis, the process by which yeast and humans and everything in between makes gametes. More remarkably, and this is partly why Mendel is revered by everyone, he didn't just make 2 astonishing discoveries. He invented a method of making them. And Mendelian methods are essentially, to this day, the foundation of the entire discipline of genetics. So the core principles that were at the foundation of Mary Claire King's work here at Berkeley to study the fact that BRCA1 causes hereditary breast cancer, the core principles of that analysis were established by Gregor Mendel. Before diving into Mendel's work, which of course was done with Pisum sativum, the edible pea, and discovered principles that apply to Homo sapiens, the thinking human, I need to remind you that, of course, we are both diploid. Allow me to pause briefly and make very sure that all of you understand what this term means. The nucleus of a female human cell in G1 before DNA replication, so a neuron, a fibroblast, non-dividing, contains 2 complete copies of the human genome. One from mom, or mom if you're English, and one from dad, which I think in England is also called dad. I want to emphasize that being diploid is not necessary for the viability of a cell. For example, Saccharomyces cerevisiae, the budding yeast, which gives us bread and wine and beer, lives perfectly happily as a haploid. Inside its nucleus is its complete set of instructions for how to build itself. Inside our nuclei, however, are two complete sets of instructions for how to build us. So in fact, humans and plants are diploid with several interesting exceptions. For example, bread wheat, in fact, is hexaploid. And we'll talk more about this later in the class. Now, we can talk at considerable length about why that's useful. But I do want to point out that there are relatively few, if any, more than diploid mammals. To the best of my knowledge, there's only one clearly characterized tetraploid mammal. So 4 copies of the genome per nucleus. Does anybody know what that is? Here's a bit of trivia. It's the vizcacha, the Peruvian mouse. It's about this big. It's pretty amazing. I will show you a movie of it later in the class. There is a correlation between ploidy and size, which is why the domestic mouse, Mus musculus, is this big, and the vizcacha is this big. Okay, so that line and that line represent 2 homologous, we will get to that term again in a second, chromosomes number 1. This is from mom, and this is from dad. Let's scoot on over to the 9th smallest chromosome, 9. This one is from mom, this one is from dad, and so on and so on. I understand all of you understand, and that was the purpose of this exercise, because it's about to get quite technical, and I didn't want to get into those technicalities without making sure you understand what diploidy means. Your textbook contains several nice pictures representing the relationship between the life cycle of the organism and ploidy. So this is the life cycle of a plant, and this is the life cycle of a human. The bottom line is the gametes that plants and us produce are haploid. The adult organism, which results, of course, from the fusion of 2 gametes, is diploid, and that's true of plants and of humans. You probably all know this already from having taken biology in high school, so I'm not going to spend any time on this whatsoever. I do want to spend a little bit of time on this slide, because I suspect most of you don't know this, but I think most of you should. The fact that we are diploid has a number of interesting and important consequences. So for example, every single one of your cells, except erythrocytes, which of course are anucleate, contains 2 copies of every single gene. Now, strictly speaking, this is true for the ladies in the audience. The gentlemen, of course, are hemizygous for the X, but that's... Furthermore, with only a few hundred exceptions that I will get to at the very end of my section of the class, both copies of each gene are active, namely, they do their thing. So your mother's copy of a gene and your father's copy of a gene are both active and are doing their thing. Now, this seems strangely redundant. I mean, you don't have 2 iPhones. I mean, I suspect some of you do. Hello, hello. But it's just a fact, and one with many significant consequences. Here's one of them, particularly relevant in the world's greatest melting point, the United States of America. January 5, 2012. Here's a child, Kyle, and he's mixed race, and he needs a bone marrow transplant, so he needs a matched donor. And like many people of mixed race, it is harder for him to find such a donor. Just a few months ago, writes the newspaper, 4-year-old Kyle Crawford was a healthy little boy, but on a family trip to Disneyland, he woke up several times with blood on his lips. He has aplastic anemia. In just a few weeks, he needs a marrow transplant, but because he's mixed race, Kyle's chances of finding a good match are slim. He will also definitely need a donor who is mixed race. This is a consequence of humans being diploid and several other things. And I will, in fact, explain them to you, but I thought this was a good moment to bring up the fact that you can register to become a bone marrow donor. It's a painful but completely safe procedure. The registration is completely painless. Giving bone marrow is not pain-free, but is completely safe, and you can save somebody's life. You should particularly consider doing this if, like Steve Jobs or Barack Obama or Tiger Woods, you are of mixed ancestry. In that case, you are uniquely valuable because somewhere in the world or in the United States, there could be, to parse back to these, a Syrian American, a Kenyan American, or an Asian African American whose life you could save. Literally, this is not hyperbole. This is a binary fact. This has to do with principles of Mendelian genetics, I will explain. The cell biology of immunological identity, I will explain briefly. And the genetic history of humanity, which I will also explain, which is quite remarkable. But before that, this cannot wait. So I encourage you with all my heart, especially if you are of mixed ancestry, to register to become a bone marrow donor. You could literally save somebody's life. With that, back to Versuche über Pflanzenhybriden, studies on plant hybrids. There are many reasons this paper is beautiful. His prose is marvelous. Those of you fortunate to be able to read and understand German, this is the only German passage in the lecture. You should read the original. His prose is wonderful. He read Leeuwenhoek's study showing that only male rabbits contribute to progeny. He read Buffon's attempt to mate a wolf with a dog, which provided the wolf with a healthy meal. And he said, those who survey the work done in this department will arrive at the conviction that among all the numerous experiments made, not one has been carried out to such an extent and in such a way so as to, first, make it possible to determine the number of different forms under which the offspring of the hybrids appear. 2, arrange these forms with certainty according to their separate generations. Or 3, to ascertain their numerical relation to each other. What is Mendel proposing to do? Well, he's first of all proposing to determine what kinds of offspring do we get. Yellow, green, round, wrinkled, purple, white. Second, did they occur in the parental, the offspring, or the grandparental generation? The P, the F1, or the F2? 3, he's proposing to count the ratio of offspring within different phenotypic classes. Believe it or not, before this peasant's son, working in obscurity in a Moravian monastery, proposed this, nobody had the idea to do that cleanly. It was his invention, this method. Today, you learn this in middle school. Back in the mid-1860s, this was revolutionary. In fact, so revolutionary that, as all of you know, Mendel died unknown, and his paper was essentially ignored for 35 years until this was rediscovered and the world went, oh. I need to walk you briefly through a bit of plant biology. The biology is relevant here because Mendel used it to great advantage. You can take a seed, and I know this sounds pathetically trivial, but it's an important fact. If you want to breed dogs, you have to have a dog kennel. If you want to breed anything else, you need a space to do that. If you want to breed plants, you need some soil, some water, and some sun. Plants are an experimentally tractable model organism that are easy to work with logistically. And in fact, the subsequent history of biology, whichever model organism you pick, the plant Arabidopsis thaliana, the fruit fly Drosophila melanogaster, the budding yeast Saccharomyces cerevisiae, the roundworm Caenorhabditis elegans, or the mouse, Mus musculus, every single model organism was picked because of some aspect of its biology that makes it easy to work with. You cannot have a situation where the wolf eats the dog. It's just not a tractable system. No, don't eat it, mate with it. I mean, so you can take a seed and put it into soil and water it, and it will grow a plant. And that plant will make more seeds. No. So I will give you a Mendel checklist. For everything that he did, I will tell you what he did and why he did it. And I am expecting you to memorize that slide, please. So I will ask you on the midterm, why did Mendel do a certain something? And you are expected to tell me. He chose to work with plants. Why? Because they don't eat each other. And they, well, some plants do, but the garden pea is relatively benign. Thus, you can grow it in laboratory settings, which in Mendel's case was his garden in his monastery. Oh. Second, plants, at least most plants, reproduce via, please write this down, normal sexual reproduction. Why did I say most plants? One of the biggest tragedies in the history of science occurred when Mendel sent his data, even before he wrote the paper, to the foremost authority of his time. And the letter is so polite, it makes me cry. Dear foremost authority, your humble servant from the middle of nowhere is apologizing sincerely for troubling us. Like, oh my God, man, you're Mendel. But he didn't know he was Mendel. And the authority reads the paper, does not understand it, and says, look, I don't understand any of this, but I think you're wrong. Here are some seeds of my favorite plant. Repeat your experiments with my favorite plant and come back when you're done. And Mendel spent his dying days doing that and failing. The seeds he received was of a plant species that does not reproduce via sexual reproduction. It undergoes something called apomixis, essentially parthenogenesis. Mendel was unable to reproduce his findings because that stupid plant doesn't follow Mendel's laws. But nobody could have known it at the time, including the authority. Teachable moment. If you get a letter from the middle of nowhere with a finding, do not send that person your materials. Read the letter carefully. Think about it. You have a choice of how you enter history, as the person who ignored the next big thing or the person who helped the next big thing make happen. Okay? The garden plant, Pisum sativum, reproduces via normal sexual reproduction. It makes haploid gametes. Those gametes fuse, and the fusion of those gametes is a diploid entity that produces the plant. That plant that produces gametes, which are haploid, which fuse, und so weiter und so fort, which is German for etc., etc. And here is a zoom-in picture of the beautiful flower of the garden pea. Now, Mendel mastered the challenging art of artificial fertilization. To cross two plants, you don't just put them next to each other and say, hi. You actually have to physically transfer the gametes of one plant to the flower of the other plant, such that this fertilization can occur. Why do this? Because the garden pea, as well as many other crop species, for example, corn, please write this down, naturally selfs, S-E-L-F-S. Its natural mode of sexual reproduction is to cross to itself. So it essentially, using incredibly non-plant-centric nomenclature, makes eggs and sperm. They're not called eggs and sperm, of course. These are plants. And then those gametes produced by the same plant fuse to form the diploid. So the natural mode of reproduction of the pea and of maize, probably the second most important plant species on Earth, second only to rice, of course, selfs. Mendel picked the pea because he could do controlled self-crosses. To do a self-cross in the pea, you do nothing. You just leave it there, and nature will take her course. To do an outcross, you artificially transfer gametes from one plant to the flower of another. Mendel bought a large number of seeds from pea seedsmen and grew the plants and was excited to discover that they differed in a broad range of characteristics. There were white-flowered and purple-flowered, wrinkle seed and round seeds, yellow seeds and green seeds. Where did they come from? Were they generated by the greater Brno Society for Targeted Mutagenesis of the Pea Genome? No, that technology was not invented until 2011. They were naturally occurring. Mother Nature produced them, and pea breeders preserved them. He picked the pea further because there are naturally occurring variants. So you can study the inheritance of differences. Imagine if all peas were green. So Mendel's work would look like this. I took a green pea and I crossed it to a green pea, and all the children were green. And then I selfed the green children, and all the grandchildren were green. And then I continued this silliness. You need to have varieties to work with, things that differ. So in Mendel's case, he could simply go to local seedsmen and buy seeds that differed. You know, people bred white flowered peas and purple flowered peas, and he just bought them both. I need to introduce you to a poorly explained and consistently misunderstood term, a pure breeding line. Mendel, in fact, established a large number of pure breeding lines. Please listen carefully, understand, and remember as best as you can. He wanted to ensure that the plants he started with exhibited a trait of interest, for example, flower color, consistently. What does consistently mean? It doesn't mean through the life of the plant. It means in its progeny and the progeny of its progeny. How do you do that? He relied on the wonderfully useful feature of the pea plant. Its natural mode of reproduction is self-pollination. This means he could take a plant with purple flowers, cross it to itself, take its purple children, cross them to themselves, etc., etc. And after multiple rounds of this, which fortunately in plants is feasible because they don't eat each other or run away, you obtain, quote, a pure line. What does that mean? It means an organism that when crossed to itself, or an organism just like it, produces progeny that are exactly like the parent in the trait of interest. So a pure line of purple flowered peas is this. It's a bunch of plants, you cross them to themselves or to each other, all the children have purple flowers. Take those children, cross them to themselves or each other, all the grandchildren have purple flowers, and so on and so on and so on. For each character, for example, flower color or seed shape, he generated two such pure lines, one exhibiting form 1 with purple flowers and one form 2, white flowers. They were grown separately. Why was he able to maintain them separately? Why didn't they cross-pollinate? They don't. That's not what the pea does naturally. It naturally self. I'm a pure line. Now we can say, of course, that the reason making pure lines is useful is because making pure lines homozygoses the genome. To remind you, RP65, the Leber's congenital amauropathy gene, there's chromosome 1 from mom and chromosome 1 from dad, and there are two alleles, forms of the gene on chromosome 1. This is the one from mom, this is the one from dad. If those two forms are the same, you're homozygous. The boy you saw treated with the virus, he was homozygous for the RP65 gene. His parents, however, were heterozygous. They were carriers. How do we know this? Because it's autosomal recessive. Take a male parent, pure breeding, purple, cross it by artificial fertilization to a female parent. Obtain first-generation seeds and see what happens. So he crossed two pure lines. Why? To get a hybrid. Remember the study of the paper. Studies on plant hybrids. Okay, the hybrid has been obtained. Let's go rest. No, the work has just begun. He took that hybrid plant, the one that's the child of a purple and a white flower, the ones, and I'm about to just disclose an important fact, whose flowers are purple. All the children from this purple on white cross were purple. He was stunned to discover that when those purple children were crossed through themselves, they produced both purple and white progeny. This was pretty stunning because he realized that this means that the whiteness of grandma did not disappear. It just, and here I'm going to use Mendel's own word, receded for one generation. The first one, only to then reappear in the second one. So Mendel selfed the hybrid, the F1 generation, to see what would happen. He had no bias as to what would occur. This was an open investigation. He was stunned to discover that the whiteness comes back. Perhaps this is an artifact of flower color. He was a brilliant experimentalist, and he worked hard to reproduce his findings with other characters. So he repeated this experiment on 7 different character sets, shown here. He made pure lines for the different forms in each case. He crossed them, got the hybrids. In all cases, the hybrid looked like one of the parents, not the other. In all cases, he self the hybrid. And in all cases, the second generation showed both grandma's and grandpa's traits. He then realized he discovered something deep and general. He correctly deduced that the notion of blending inheritance in this specific case just doesn't apply. The hybrid, F1, looked like one of the parents, but was genetically different. The parent, when crossed to itself, the pure breeding line, I'm a pure breeding line, produces progeny that look just like it. However, when the F1 hybrid is crossed to itself, it produces progeny that look both like its dad and like mom. So latent in the hybrid, there lies some entity, a factor, the one it got from mom, that recedes into the background for one generation and then reappears. Mendel then coined the term dominant and recessive to refer to purple and white respectively and to all the other traits that behave like flower color in his crosses. He really invented that term. It then got better. Because he was a student of Christian Doppler and was carefully trained in mathematical analysis, he proceeded to count the ratios of those that look like grandma and those that look like grandpa. And in all cases, it was very close to 3-to-1. And then, through analysis, I unfortunately don't have time to explain, but if you take an upper division genetics class like MCB 140, you will learn all about this. Because he was brilliant and he was trained in quantum analysis, he discovered the first law. I'm just going to give it to you and then explain how he got to it. This is not the definition in your textbook, and this is the definition I'm expecting you to know. Half the gametes made by an organism heterozygous for a given gene locus carry one allele of that gene and half the other. Gamete, a haploid cell that fuses with another gamete in the sexual life cycle to form a diploid zygote. Heterozygous, carrying two different forms of a given gene. Locus, the place in the genome where a given gene lies. Allele, a form of a given gene. Mendel introduced simple nomenclature. He was the first one to do so. Big A for the dominant, small a for the recessive. Mendel invented it. So what you learned in middle school, cursing your life, was invented by Mendel in the 1860s. So what's going on? Well, we now know, of course, that organisms carry in them two separate entities. We call them alleles of genes that determine the phenotype of an organism for a given character. A given gamete contains only one allele of a gene. A hybrid plant is diploid, and while resembling only one parent, is in fact, of course, heterozygous. I'm flying through this because you learned this, I think, already twice. Once probably in middle school, once in high school. But when the heterozygous plant makes gametes, half of them contain the dominant allele and the other half the recessive one. And then elementary laws of probability give us this beautiful 3-to-1 ratio. So I flew through this at rocket speed because you all know this, but I want to pause for this. There is this son of a peasant working alone in Brno in the 1860s. And here's this diagnostic testing that resonates to this day. This is Christina Applegate. She's a brave woman. She is a BRCA1 mutation carrier, and she went public with it and the fact that she had the surgery. And this raised the profile of this issue. So it's remarkable, isn't it, how studies on model organisms in relative obscurity resonate and save people's lives. Why is Mendel's first law true? What biological aspect of living things explains the fact that this happens? The dynamics of meiosis. Please pull out your clickers. I need you to find the wrong statement, and for this one, because it's relatively straightforward, I'm going to give you 60 seconds, not a minute and a half. But I'll read this first. As promised, I'm asking you about something you saw last time, which is wrong. A, DNA replication must occur for the entity shown on the right to be formed. B, once all chromosomes in the cell have assumed the position you see here, the metaphase-anaphase transition can occur. C, the mitotic spindle separates the sister chromatids and parses them into the daughter cells. D, the sister chromatids are genetically distinct. E, during telophase, the sister chromatid starts decondensing to become interphase chromosomes. Okay? right. Go for it.
Yeah.
20 seconds.
You're gonna get all these eye clickers thrown at you.
Comes with the territory.
Just keep on pressing until it works.
All right.
587, I think.
There's not 587 people here. Let's see what you said. Yeah. Of course, they're identical. They're the products of DNA replication. Good for you. Why did I just tell you this? Why did we just spend all this time on this? Well, because, go away. This highlights a critical difference between the two types of cell division that we've studied. The purpose of mitosis, the raison d'être, is to generate two daughters, DNA replication, that are identical. DNA replication occurs, and then two genetically identical copies of each chromosome, sadly known as sister chromatids, are parsed into daughter cells. The purpose of meiosis is to make a haploid from a diploid, one complete genome per cell. There's DNA replication. But then for each chromosome, 1, 2, all the way to the X, the copy from mom is sent to one cell and the copy from dad to the other. Furthermore, unless you're a Drosophila male, at this point it's no longer correct to even call them chromosome 1 from mom and chromosome 1 from dad. And why we shall discuss next time. Here is a nice picture from your textbook which reminds us that for any given pair of chromosomes, this is chromosome 1 from mom and this is chromosome 1 from dad, genes lie as follows. Here's a gene and here are its two forms. And what will happen to these during meiosis? What will happen to these during meiosis is the following. DNA will replicate and the homologs, the one from mom and the one from dad, will pair. And they will pair in such a way such that the homologs then will go to separate cells. So chromosome 1 from mom, chromosome 1 from dad will separate like this. And then in the second meiotic division, of course, the sisters, which I haven't even mentioned yet, will go to separate cells. But from the genetic standpoint, that's at this point irrelevant. What matters is Mother Nature has managed to parse the two homologs with the two different forms of the same gene into the left gamete and the right gamete. So Mendel's first law is true because during the first meiotic division, something remarkable happens. In mitosis, of course, DNA replicates and two identical copies of each chromosome, the sister chromatids, go to separate cells. In meiosis, homologous chromosomes, so related but not identical in sequence, find each other, pair at the metaphase plate, and then the cell separates the homologs into separate cells. And this means that for every single gene locus, one gamete gets one allele and the other gamete gets the other allele. And that's why Mendel's first law is true. Mendel then repeated the experiment. He's a very hard-working man. This was a very difficult experiment to do, but with pure lines differing from each other in two characters. Why? Because he wanted to know if the first law would still work. This is Professor Jasper Rhine. He founded dog genomics, more or less. And why am I telling you this here in Berkeley? Because subsequent work, which remarkably, in a perfect coincidence, I was going to talk about this anyway, is on the cover of the current issue of National Geographic. I didn't talk to them about aligning their publication schedule with teaching Bio 1A, but they did. And what you will discover is that the principles of inheritance of the Bichon frisé, or the Australian Terrier, or the Golden Retriever, are beautifully explained by Mendel's second law. And I will teach that to you on Monday. But before I can do that, I can tell you what Mendel's second law is. He did a dihybrid cross. Bear with me, we're almost there, and there's a cartoon at the end of this which will reward you for your patience. Parental lines, I promised, pure breeding, differing in 2 colors. Round seeds, yellow color one. Wrinkle seeds, green color the other. Cross them. All the progeny look like one plant. They're round-seeded and yellow. Self, the hybrid, and yes, of course, you remember, he observed a remarkable 9:3:3:1 distribution of phenotypes. He then did analysis I cannot do justice to in the time I have, to simply show that therefore this means that when you do a cross like this plant makes 50% gametes with the R allele and 50% gametes with the recessive allele. Furthermore, for the other gene locus, it makes 50% gametes with the dominant allele and 50%, bad professor, bad professor. Sorry, if I had the Men in Black device, I could just erase your memory of this with the Y, the lowercase y. The law of independent assortment, the formulation of which I need you to know. Alleles of one gene follow the first law, independence of alleles of other genes. There's a really cool exception to this. We'll talk about it on Wednesday. But for now, this is what I need you to know. What do you mean follow? Follow means get distributed or assort to the gametes. I need your all-out attention for the next 60 seconds so I can explain why the second law is true. It's very deep and very cool. Recall that law 1 says that for each pair of homologs, the one from mom goes one way, the one from dad the other. What about 2 different homologous chromosomes? The long one and the short one. The one with the seed color and the one with the seed shape. It is clear that homologs form pairs and align without consulting each other. The 2 arrangements are equally likely. Let me zoom in on this region. This is meiosis 1. It's not like the long chromosomes align and say, hey, short ones, is the mom one going this way and the dad one going that way? Okay, then we're going to do the same thing. Similarly, the short ones don't consult the long ones. They do their own thing independently of the other chromosomes. This means that this arrangement, where R goes this way and small r goes this way, big Y goes this way and small y goes this way, is equally likely as this one. The 2 homologous chromosomes follow Mendel's first law independently of each other. There's a wonderful piece of stepped art, and I know Professor Meehan asks you about this as well, that walks you through the kinds of gametes that you can generate using this process. Please go ahead and click through this. It'll help you understand like that. Last thing, the last 15 seconds, and with that you're free to go. Thank you for your patience. If you have no particular plans this weekend, go see the Charles Schulz Museum in Santa Rosa. It has wonderful idiosyncratic cartoons, such as here, talking about dogs. Snoopy's writing a novel. It goes, though her husband often went on business trips, she hated to be left alone. I've solved our problem, he said. I've bought you St. Bernard. Its name is Great Reluctance. Now, when I go away, you shall know that I'm leaving you with great reluctance. She hit him with a waffle iron. Have a nice weekend.